All right. My name is Ben Bateman. I'm the team lead for iGEM WLC-Milwaukee. And I'm going to be talking to you about our project a little bit today. This isn't our actual presentation slide deck, just so you know, so it might be a little ugly. So we think that SynBio is awesome, but there seems to be a stigma that it's very difficult or impossible to get introduced to. And when we were thinking about the foundations of synthetic biology and how we can get more people introduced to it, we thought that the best time to do this was during the gene regulation topics in introductory bio courses. However, this is notoriously one of the most difficult sections of these courses. It's dense, it's conceptual, and as a lot of it is personified, when in reality, that's not really what's happening. We say that the repressor binds, that the operon is turned on, but where does the repressor bind? Where does it come from? What does turning on mean? We also notice that there's no easily implemented hands-on lab kit to teach gene regulation, and we wanted to change that. So before we totally committed to this, we wanted to get a better idea of what college professors and high school teachers thought about this idea. We interviewed a ton of educators to get their thoughts and philosophies on hands-on experiences, and it turns out that they love them and they gladly shared their thoughts with us. First, every good lab kit has an even better lab manual that includes tons of background information, how to use the kit, supplemental exercises, and other things of that nature. Second, the kit should work a majority of the time. There's nothing worse than talking about something in lecture and then doing a lab about it just to have the lab not work and have nothing to bolster what you talked about. Third, it should also be able to be scaffolded to different levels of understanding. So this was super helpful in designing our project, and we folded their feedback into the base layer. After this, we wanted to get an idea of how different students learned. So we did a few lab demos where members of our team got to teach about DNA and SynBio firsthand. In this, we immediately noticed that not every student had the same background in the information. We had to be sure to implement this idea into our kit and into our lab manual. So how do you go about solving a problem like this? Well, we wanted something with instant gratification, the type of science that you get really excited about. And we thought that the best and most safe way to do this was to do a color change. We thought that a color change would get people excited about gene regulation. We also wanted some terminology that students would be familiar with. The ubiquitous system of genes that is used to kind of teach gene regulation is the lac operon. And additionally, its components are really well documented and very well understood. So we found that the major product of the lac operon, beta-galactosidase, would actually induce a color change when exposed to the substrate XGAL. However, beta-galactosidase is the end goal of the lac operon. To an introductory bio student, that's the easiest part. The regulatory components like LAC-I, the cap binding site, LAC-O, are much more difficult to understand. We thought we could delete regions of the lac operon to show how the regulatory components affect the overall function. And that is our project's goal, to create constructs of the lac operon with different regions removed, allowing for quick determination of whether or not these structural genes are being expressed. So how did we do it? We started by trying to get the whole lac operon into PSB1C3, and this proved to be fairly challenging. We designed LAC-I and LAC-Y biobricks and were able to clone them into PSB1C3 and transform them into E. coli. But getting LAC-Z in there as well didn't seem to work. After many tries, we concluded that this just wasn't going to work with the resources that we had. And we believe because LAC-Z is too big of a gene to insert into PSB1C3 along with LAC-I and LAC-Y. The resulting fragment to insert would have to be longer than PSB1C3 itself, which means we were really inserting the plasmid into our DNA fragment. We didn't give up. We tried to find different genes that, when expressed, induced a color change, and we found BLA and ICM. BLA expresses beta-lactamase, and ICM expresses indole 3 carboxylic acid monooxygenase. Beta-lactamase from BLA turns red when exposed to nitrocephin, and iCM turns purpley blue when exposed to indole-3 carboxylic acid. So we obtain plasmids with these genes and the lac operon's regulatory components, and we transform them into E. coli. And then it was time to test, and it worked, at least the BLA construct did. BLA turns the culture red when exposed to nitrocephin in the conditions expected for lac operon functioning. That is high concentration of lactose and a low concentration of glucose. iCM did not work. So now that we had a fully functioning construct, we could start to break things. We aimed to do 6 mutations. We wanted to mutate the LAC-I promoter, LAC-P, the cap binding site, the LAC-I open reading frame, LAC-O, and the LAC-Z ribosome binding site. So we did the mutagenesis and we sequenced the results and found that we had 3 sequencing verified mutants and 3 mutants that seemed to be working, but the sequencing quality wasn't good enough to actually confirm that they were there. So we tested them again. So the LAC-I promoter mutation was functioning as we expected. Without LAC-I expression, LAC repressor protein will not bind LAC-O, leading to expression of BLA regardless of the environmental conditions. The LAC-P mutation was also functioning as expected and sequence verified. Without LAC-P functioning, BLA will never be expressed regardless of the environment. The cap binding site mutation is a little weird. Its mutation is sequence verified, but it doesn't seem to be functioning as well as we'd hoped. We should be seeing a little bit of color change in the only lactose one, but we aren't seeing any, so we're still working on that. We changed some codons in the LAC-I open reading frame to be stop codons, and this mutation was not sequence verified, but it is functioning as we expected in at least this colony from the transformation. So without LAC repressor production, BLA expression is expected regardless of an environment. The LAC-O mutation is also not sequence verified, but it does seem to be functioning as intended. Without a place for the LAC repressor to bind, continued expression of BLA is expected, leading to color changes in any environment. And finally, the LAC-Z ribosome binding site mutation is also not sequence verified, but it does seem to be functioning as intended. Without the ribosome binding site, BLA cannot be translated into a functional protein, and such there is no expected color change regardless of environment. So now that we had a functioning kit, it was time to turn our attention to the lab manual that our experts that we interviewed stressed was so important. Our team worked to ensure that our lab manual is top-notch. We included how to use the kit, what reagents you might need, safety information, any prep work that had to be done, resources for teachers like concept checks and worksheets for students, any troubleshooting tips we include in there, and then we also point to other well-established educational tools. In my opinion, best of all, our lab manual is divided into 3 different sections based on understanding. So one is for the high school level, one for introductory college, and one for upper level biology. Unfortunately, our team hasn't put much thought about the logistics of this project into an actual like marketable, purchasable lab kit. Right now, our project can be very easily implemented into institutions with basic microbiology lab equipment, but other more under-resourced institutions might struggle. Most significantly, nitrocephin, the substrate, is very expensive and would inflate the cost of our kit further than I am comfortable asking an underserved institution to pay. To combat this, we can try to find a gene that expresses that when expressed induces a color change using a cheaper substrate. Additionally, institutions with no means to culture bacteria would struggle implementing our kit. To do this, we're considering putting already sterile antibiotic treated media into the kits for them to use. That way, these institutions will only have to provide an incubator. So that's our project. I really hope you enjoyed this. I'm very excited to answer any questions you might have and also hear your feedback. I also have a couple questions for you and your team, so I'm very excited to meet with you, and I'll see you then.